You'll see that we're now dressed for the occasion, evening meal later. So we have our Virkon close at hand. We're working inside a tray for fear of any spillages. If there are any, then they're well and truly contained, and we have our Virkon emergency and disposal as well. That's right, isn't it, Mary? We have our solutions, cheek cells in saline, nice little number, and we have a cheeky little saliva mixture here that again we're hoping to extract DNA from those two sources. And we're ready then, I think, Mary, not to transpose the right amount of materials into the appropriate vessels.
Yeah. Now what we're going to do is we're going to concentrate the cells, and we're going to use a centrifuge, a mini centrifuge, to do this. So what you're going to do, John, is handling your own materials for health and safety reasons. You're going to transfer an equal amount of saline with your cheek cells into two of these little plastic tubes. After the spinning, the cheek cells will be down the very bottom, the very tip of the tube as a pellet. And you're also going to transfer an equal amount of your saliva into two other tubes.
Okay. So for balance purposes, all four tubes need to have the
same amount,
okay. Well, we'll endeavour to do that. Make sure to put them in the one. So that's from the saliva.
And for the saliva, you're looking around about 0.5 ml in each tube. As long as it's an equal amount, it doesn't really matter.
Okay, we'll pop the top on for the time being. Crack.
We find these little foam racks very useful for holding these very small tubes.
Okay, there's my second sample of saliva. I think that's not far off the same. I'm going to pop that into out the way.
Mrs. John, thank you for putting your pipettes straight into Virkon immediately afterwards to kill any microbes.
So the same again for the salt solution. Give that a bit of a whisk up. Click. And the final one, a bit tedious, but sure.
And in a classroom situation, as long as each student transferred the same volume into their tube, then that's fine. They don't need to do double tubes in the classroom.
So we've got two tubes just to duplicate.
Because there's only you,
and we've got them labelled up with my initials, and in this case CC is for cheek cell source, and JB Sal is for a saliva source. So we're going to pop those into the microcentrifuge.
Yep, if you pop them in, and just make sure that the equal tubes are opposite each other on the centrifuge.
So we've got that, and that, and that.
And then
okay.
just put the top down. And